I'm sure most of you have heard of GMO organisms, genetically modified organisms. Um, these organisms are made through a process of genetic engineering. Um, the reason this is possible is because DNA is universal to all living things, meaning it is found in all living species. Why is that? Well, the first organisms that evolved probably had DNA, right? So DNA evolved in the tree of life down here, and everyone else who descended from that early life also have DNA. But because of that, we can take DNA from one group of organisms and transplant it into another, and they will produce the proteins we're interested in. Remember, DNA, if you remember the central dogma of molecular biology, codes for RNA, which codes for protein. Protein is made using ultimately DNA as a blueprint. So, one way to to do this, to get the proteins we want or to get the DNA we want, to make a bunch of copies of DNA, is DNA cloning. And we do that with bacterial plasmids. So if we look at a bacterial cell, they have a ring of DNA known as a nucleoid. But they also have smaller bits of DNA known as plasmids. Plasmids sometimes can be shared between bacteria, and often they contain genes that are only used in special circumstances, like during a hard drought, let's say, or some sort of tough conditions. Those plasmids might turn on, and the genes might be expressed into protein. But scientists can take a plasmid and they can insert DNA into it. So let's say you have a bacterial plasmid right here, and you want to make a bunch of copies of DNA from a fish. So you take DNA from a fish that you're interested in, and you cut this plasmid and you glue the fish DNA in. And then when that thing copies itself, it's going to make a whole bunch of copies that also contain the DNA you're interested in. So you can get, I'm just going to very crudely draw that over to the top there. There we go. All right, so we get lots of copies of what we're looking for, okay? This is known as recombinant DNA because it has DNA from two species. It's combined. And so what you can do is you can put this together, put that bacteria into a test tube full of nutrients, and it will divide over and over and over again. And you can get lots of copies of the DNA that way. Or you might collect the proteins produced this way. Because remember, DNA to RNA to protein, these bacteria are going to be pooping out lots of different proteins because of that new DNA. And maybe it's the proteins you're interested in. So you can see that in this diagram, where they take DNA from a species of interest right here, and they inject it into, I shouldn't say they inject it, but they splice it into the DNA of the plasmid of a bacterium. So here's the plasmid. They put the gene of interest from another organism in, and now they let it divide. It's now a recombinant bacterium. It has the DNA of two species in it. It divides. Now, a couple things you might do with that. You might later on take that gene out of there and put it into other organisms. So, for example, corn. Corn has a gene in it from a species of bacterium. Well, I shouldn't say all corn, but corn that we often grind into feed or perhaps get at the grocery store has a gene in it from a bacterium that we've placed into it. What that gene does is it makes the corn resistant to things like caterpillars. Caterpillars eat the corn. That gene makes a toxin that kills the caterpillar, but it does not harm humans. So we've done that before. We could also put a gene into bacteria like this that makes that bacteria able to eat oil in an oil spill. Maybe that's another use for this for a recombinant bacterium. Okay. Another thing that could be done is, instead of harvesting the DNA or using the bacteria to do something new, we might actually collect the proteins instead. So some real life examples here. We can do this. We can put a gene into a bacterium, a human gene into a bacterium, and it will produce human growth hormone. We can then take those proteins and use it to help folks with stunted growth. Or we can produce proteins that break down blood clots to help heart attack patients. So there's lots of options here, using the DNA itself, using the bacterium that we've altered to do a new job, or collecting the proteins and using those for something new, like medicine. Now, one of the ways we would do this is we would take a bacterial plasmid and we would use something known as a restriction enzyme. A restriction enzyme is an enzyme. An enzyme is a type of protein that has a very specific shape, okay? And in this case, it's like a pair of scissors that cuts DNA at a very particular spot. So in this case, this area is the spot it can recognize, and it will cut it, okay? So what happens is it cuts that spot. And notice it leaves some spots overhanging. So here's kind of how that DNA looks, right? There's the... I'm kind of drawing this over here, right? So it overhangs a bit, and this part right here is known as a sticky end. It has base pairs that are not stuck to anything yet. So let's say this is AGC, right? AGC would really like to stick. Well, I'll let you decide what would stick to AGC in my example over here. Well, maybe a T, a C, and a G, right? So if you take DNA from another organism that also has what we call sticky ends. They're called sticky because they are base pairs that don't have a partner. Remember, A, T, C's, and G's like to form partnerships, those hydrogen bonds. So they're sticky. They really want to stick to another base. You stick in the DNA from another organism into that spot, and it will glue itself in, especially once you add in DNA ligase, that last enzyme we talked about when we mentioned how DNA replicates. It will glue those covalent bonds, finishing the DNA backbone there, gluing those pieces together. Now, what you might do is you might put in a restriction enzyme, and it might cut a lot of pieces of DNA, because several pieces of DNA might have that recognition, that sequence that it can recognize, like it says up here, a restriction enzyme sequence here. So if that's the case, if it cuts several pieces, you might have to figure out which piece you want. And one way to do that is through this process here known as gel electrophoresis. So in gel electrophoresis, you put DNA into a gel, which is basically like Jell-O. And to a piece, let me draw a gel over here, crudely anyway. There it is. Up in this area, you'll put the DNA, okay? And this area is going to have a negative charge, and this area is going to have a positive electrical charge applied to it. Now, DNA has a negative charge because of those phosphate groups that have a negative charge, okay? So they're drawn towards a positive charge. Now, to a piece of DNA, a gel is like a forest. I mean, to us, it looks like a semi-solid substance, but to a tiny piece of DNA, there's lots of cavities and openings. And a big piece of DNA is going to bump into the gel a lot, and let's say you turn this thing on, you turn the electricity on for an hour. A big piece of DNA is only going to move a little ways. Whereas a little piece of DNA, or several little pieces of DNA, will move further. So you can actually separate out all the pieces that you've cut up, and then you can sample those to see which one is the piece of DNA you actually want. So now that you found it, you have the piece of, you found the plasmid that you cut, and now you want to add in DNA. And so to add in DNA, you might need to take another organism, let's say a mouse or whatever, and you find the gene you want, and now you have to make copies of it to put it into the plasmid. And to do that, you use a process known as PCR, or polymerase chain reaction. This is just like the DNA division we talked about earlier, except in a test tube. Now to do this, you put the DNA you want into the test tube, you heat it up, and they call this denaturation. That's when you break the hydrogen bonds, and you do that by heating it up. Hopefully you get to do this in genetics, when you take genetics. Then you add in the primers for that piece of DNA. We've talked about those already. You add in DNA polymerase, and you make copies of that DNA. And then you heat it up again and open it up, and you make more copies, you make more copies, you make more copies, until at the end you have a whole bunch of copies of the DNA that you want. And then you cut it with a restriction enzyme, and then let's go back here, you add it in and you glue it in to the plasmid that you've made copies of. Now, another thing that we can do, and this is a fairly new thing in the last 5-10 years, is a process known as CRISPR-Cas9. And this is a enzyme known as a nuclease, meaning it's a enzyme that cuts nucleic acids. And in this case, it cuts double-stranded DNA. And it does that with the help of guide RNA. So guide RNA is RNA that is complementary to the target gene. Once again, if the target gene was CGC, that's really messy, but that's as good as I can do. Then the RNA that's complementary to that would be GCG, right? It's complementary to that. So it would find that area, stick to it, then cut it. And one thing that we've been able to do with this is to figure out what certain genes do. So we might take a mouse and we know it has a certain gene. We'll find a piece of RNA that's complementary to that gene and we'll cut that gene and knock it out. And that lets us see what that gene actually does. So here's an example of CRISPR-Cas9. Here's the Cas9 protein. Here's a piece of RNA that is complementary to the thing we want to cut. And it comes in, it finds that DNA, and then it cuts it. And that can result in one of two things happening. One, it can cut the DNA. So if you cut the DNA, so let's look at this piece of cut DNA. We'll call this part number 1 and part number 2. Down here we can see part number 1 and 2. The cell can repair the cut, but the cell, because you cut both halves of the DNA out of there, the cell doesn't know what bases are supposed to be there. So it randomly fixes that hole, and that shuts off the gene because the protein that's produced won't make any sense anymore. Okay, well, that'll shut off that gene. The other thing you could do instead is you could cut it. So we have part number 1 and part number 2 here. But what you might do as a scientist is add in part number 3, and that's the DNA you want to be there. So the cell will repair that cut by putting in new DNA, and that might allow you to fix a gene. So you can shut off a gene or you can fix a gene. You might be able to do that. For example, they've been trying to use this to cure patients that have sickle cell disease by cutting out a piece of the gene that codes for sickle cell and replacing it for a cell, replacing it with DNA that codes for normal hemoglobin. We'll talk a little bit more about sickle cell in an upcoming chapter. So CRISPR-Cas9 lets us shut genes off, lets us repair genes. The other things we looked at earlier let us make copies of genes and put genes of interest into a bacterium to make lots of copies, either so we can get more copies of that gene so we can stick it into other organisms, or so we can collect the proteins to make medicine. Those are some really good examples of genetic engineering in action. And you might get to do some genetic engineering as an undergrad researcher here in the UCA biology department. All right, we're going to continue on with gene expression in the next lecture.